There are three parts to the cell cycle: mitosis, interphase, and cytokinesis. The cell cycle, or cell division cycle, is a series of events that take place in a cell leading to its division and duplication.
Let's get ready to rumble!
Interphase is this phase of the cell cycle in which a cell spends the majority of its time in performance preparation for cell division. There are three stages of interphase that end with a cellular checkpoint. The stages of interphase are G1, in which the cell grows and functions normally, synthesis, where the cell duplicates its DNA, and finally G2, in which the cell resumes its growth in preparation for mitosis. The process of mitosis is when a cell will multiply and create new cells. We will not survive if our cells cannot heal us from injuries, viruses, and illness.
Let's get it started in here.
Chromatin fibers break into discrete chromosomes. Nuclei disappear. Each duplicated chromosome appears as two identical sister chromatids. Mitotic spindle begins to form as microtubules rapidly grow from the centromeres. Nuclear envelope breaks into fragments and disappears. Microtubules emerge from the centromeres at the ends of the spindle. Some spindle microtubules attach to the kinetochores. Others make contact with microtubules from the opposite end.
Come together.
Mitotic spindle is fully formed and the poles are at opposite ends of the cell. Chromosomes come together at the metaphase plate. Centromeres of all the chromosomes are lined up on the plate. Anaphase is the shortest stage of mitosis. Two sister chromatids of each pair part, each becoming a chromosome. The two daughter chromosomes move towards opposite ends of the cell. Daughter nuclei appear in the two poles as nuclear envelopes form around the chromosomes. Chromatin fiber of each chromosome uncoils. Nuclei reappear. At the end, the mitotic spindle disappears. This is division of the cytoplasm. This happens hand in hand with telophase. Two daughter cells completely separate soon after the end of mitosis. Mitosis produces two diploid somatic cells, an identical copy of the first cell, with the same amount of chromosomes, both having 23 in humans, that is. Meiosis is important for many reasons. During sexual reproduction, meiosis is important to allow the children the same number of genes as their parents. Meiosis is also important for the reproduction of four haploid cells. Meiosis allows a child to have the genes of both the parents.
The nuclear envelope breaks down and the centrosomes move apart, beginning the formation of the spindle apparatus. The chromosomes condense with each pair of homologs in synapsis. Crossing over occurs between the homologs, forming chiasmata. Microtubules then attach to the kinetochores of the homologs.
Come together.
The pairs of homologs line up at the metaphase plate with the two sister chromatids of the same homolog attached to the kinetochore microtubules from the same pole. Homologous chromosomes separate and move toward opposite poles. The duplicated chromosomes end up at opposite sides of the cell and nuclear envelopes form. The cell divides in two during cytokinesis, forming two haploid daughter cells with duplicated chromosomes. In each cell, the spindle apparatus reforms, the nuclear envelope breaks down, and microtubules attach to the kinetochores of the sister chromatids.
Come together.
The chromosomes line up at the metaphase plate with kinetochore microtubules attached to each sister chromatid. Sister chromatids separate and move towards opposite poles. The chromosomes condense and the nuclear envelope forms. Cytokinesis results in four haploid daughter cells. After one parent cell goes through meiosis, four genetically unique haploid daughter cells are produced. In both meiosis and mitosis, the chromosomes duplicate during interphase. Prophase 1 of meiosis differs from prophase in mitosis because crossing over occurs between homologs forming chiasmata. The crossing over results in genetically unique cells, while in mitosis clones are produced. In mitosis, 2 diploid daughter cells form, and in meiosis, 4 haploid daughter cells form.